Hey, Dr. O here, and today I'd like to talk about epithelial cell junctions. Epithelial tissue can be found throughout the body. It acts like a covering, and it can be found lining the surfaces on the inside of the body as well as the outside of the body. It comes in two main forms. There is epithelium that acts like a lining, and there's epithelium that acts like glandular tissue. Now, lining epithelium can be found on internal and external surfaces of the body. So it could be found like in the skin, or it could be found lining an organ like the stomach or the intestines. Glandular epithelial tissue will be found within glands, and the cells that make up these glands are typically responsible for making some sort of a product and releasing it, such as digestive enzymes in the pancreas. So as we see, epithelium can do a variety of functions. It can be protective in nature, such as what we see in our skin. It can perform absorption, such as absorbing something from one area of the body and then moving that substance through itself to another area of the body. That's what happens in the lining of the intestines. It can perform filtration, which also involves moving substances from one area to another. That's what we see happening in a part of the kidney known as the glomerulus. It can also perform something called excretion and secretion, which are other forms of moving substances in and out of areas. That happens in a different part of the kidney, known as the tubule system. And it can provide sensory reception as well. So we see that epithelium is a functionally diverse tissue type. And despite these variations, the one thing that epithelial tissue must have is connection with adjacent epithelial cells as well as adjacent tissue. So let's see how epithelium makes these connections. Epithelial tissue will have what's called polarity, which means that it has two separate ends. We can think of these ends as a top and a bottom, but more specifically, this polarity involves a free surface and an attached surface. The free surface is called the apical surface. This is the surface that is exposed either to the surface of the body or within a cavity, such as the stomach or the intestines. Most apical surfaces will be smooth, but there are some that are going to have these finger-like projections called microvilli. The whole purpose of microvilli is to improve absorption. So these would be cells that are doing some sort of bringing in of substances from the outside and then moving those substances either within the cell or to some other area of the body. So we would see microvilli lining the intestinal tract as well as the tubule system in the kidneys, because that's what both of those cell types are doing. Now, the bottom surface is called the basal surface, and this is going to form an attachment to an anchoring membrane and ultimately connect the underlying membrane to connective tissue. Now, this anchoring membrane can be a basal lamina or a basement membrane, and this structure is what holds the epithelial cells in place and allows them to congregate and create tissue. Because of the polarity of these cells, each surface is going to be performing a different function. But what I'd like to talk about is how these cells connect not only to each other, but to the structures around them. So let's do that. It's important that epithelial cells form connections not only with each other, but with the surrounding tissues. So we're saying cell-to-cell junctions, but the reality is that it's cell to something else junctions that we're looking at here. And that something else could be either a cell or it could be surrounding tissue. Regardless, the function of epithelium requires that these cells fit together. And they fit together very closely, or at least maybe closely but with a little bit of looseness in there, because it's going to depend upon what the cells are doing. And it also is important that they are bound to that basement membrane in order to create tissues and even glands. Because these junctions are going to be specialized, we can call them specialized binding contact points. And within epithelial tissue, there are three different types. You have tight junctions, gap junctions, and anchoring junctions. So let's have a look. Now, as the name implies, a tight junction is not going to easily allow substances to pass between cells. Because of this, these junctions allow epithelium to act as a barrier. But even barriers can selectively allow certain things to pass, and tight junctions can do that as well. The way a tight junction is created is that you have a transmember protein that is anchored within the cell, and then part of it is outside the cell and attaches to an adjoining transmember protein found in a neighboring cell. Typically, tight junctions are going to be found towards the apical surface of these cells. So where would you find a tight junction? Well, actually, they're found throughout the body, but we can think of them specifically in places where we would want to keep the contents in one location, or at least closely control its movement from one area to another. For example, the urinary bladder, or even the intestinal tract. A gap junction does allow for a bit more space between the cells, but more importantly, it allows an avenue of communication between cells. So what are the cells communicating about? Well, they're not talking about the latest gossip or sharing stock trading tips. They're not interested in that. But what they are interested in is exchanging ions, maybe some small molecules, and conveying electrical impulses between cells. The gap junction is a small protein channel that connects two cells together. One cell contributes half the channel and the neighboring cell contributes the other half of the channel. And as we can see in the image on the lower right, they don't appear in isolation but in patches on cell surfaces. This enhances the communication between the cells. Now, the operative word with gap junctions is communication. So where might these connections be found? In cells where communication is important, such as nerve cells. Also in the heart, where it's important that the cells create a coordinated activity, such as contracting of the heart chambers. Next are the anchoring junctions, and these connections are going to link cells to cells or cells to extracellular matrix, something like a basement membrane. These junctions are going to create the structural cohesion needed to create tissues and glands. It also gives these tissues the strength to withstand mechanical stresses, so they're going to be pretty strong. There are three types of anchoring junctions. We have desmosomes, hemidesmosomes, and adherens junctions. Now, before we get into this, I want to point out something on my PowerPoint images here. Now, what we can see with these images is that we have a plaque, which is what this oval is in all of these pictures, and projections going into the cell. That's what we see here, and on these two. And then there are projections coming out of the cell. So here they're blue, here they're green, here they're green, and here they're blue. Now the projections going into the cell, these are anchoring the junction to the cytoskeleton of the cell. The projections coming out of the cell are anchoring the cell to either another cell or some other structure, such as the basement membrane. And you'll notice that some of the internal anchors are red, and some of the internal anchors are brown. This tells us that we have two types of internal protein anchors. You'll also notice that the cell-to-cell anchors are blue, and the cell-to-basement membrane anchors are green. This tells us that different proteins are anchoring cells to cells than anchoring cells to basement membranes. So let's have a look at all this. Desmosomes are going to link cells to cells. The internal anchoring protein is going to be called the intermediate filament, and it's made up of keratin. And keratin is a strong protein fiber that's used to make up our hair, our skin, and our nails, so it's pretty tough stuff. It's also used right here for the purpose of connecting the desmosomes within the cell. So keratin is going to be what attaches the plaque to the cytoskeleton within the cell. And we can see that it is right here with these red lines. Now the protein that's going to connect one cell to another cell is called cadherin. This is a transmembrane linking protein. So as the name implies, transmembrane is going through the plasma membrane, and linking protein means that it will actually form the attachments between the two cells. Now if we look at the general placement of these two desmosomes, we can see that they're roughly in the upper part of the cell towards the apical surface, but still within the mid area of the cell. Hemidesmosomes are going to link cells to extracellular matrix such as a basement membrane. The proteins that link the junction to the cytoskeleton are also going to be keratin proteins. That's why I show them in red. But the proteins that are going to link to the basement membrane, these are going to be integrins. So the integrins are going to be the transmembrane proteins that create the linkages. These junctions, these hemidesmosomes, these are going to be found on the basal surface of the epithelial cell. Our final anchoring cell junction is the adherens junction. Adherens junctions have some versatility to them. These junctions can either link cells to extracellular matrix, or they can link cells to cells. The internal protein that's going to link to the cytoskeleton is going to be actin in both cases. But the other protein will depend upon what it's linking to. So if the junction is linking the cell to a basement membrane, then the protein will be integrins, just like what we saw with the hemidesmosome. But if the junction is linking to another cell, then the protein will be cadherin, just like what we saw with the desmosome. So if this junction is linking cell to cell, we're going to typically find it in the upper region of the cell towards the apical surface, but still within that mid-range. If the junction is linking the cell to the basement membrane, this will typically be found on the basal surface of the epithelial cell. Now these particular junctions can be found either in isolated patches between cells and what they're being linked to, or they can be found in belt-like structures within the cell and actually cause the cell to change shapes a little bit. So let's review these anchoring junctions, the proteins, and what they're linking to. Just put it all on a chart here. So we started off talking about desmosomes, and we said that the internal linking protein for desmosomes was keratin, and that the transmembrane linking protein was going to be cadherin, and that desmosomes' job are to link cells to cells, and that we would typically find these in the upper mid-range of the lateral cell surface, not quite up to the apical surface, but more towards the apical surface and the basal surface. The next anchoring junction that we talked about was the hemidesmosome. We said it also had keratin for its internal linking protein, but its transmembrane linking protein was going to be integrins, and that this particular junction was going to link the cell to extracellular matrix, such as a basement membrane. And as a result of this, its location would typically be on the basal surface of our epithelial cell. Then we moved into adherens junctions, and we actually talked about two different ones. We said there was some variability here. The thing that they have in common, regardless of which one they are, is that their internal linking proteins are both going to be actin, but their transmembrane linking proteins are going to be different depending upon where they're found. So if we have an adherens junction that has a cadherin transmembrane protein, then we can expect that this is going to link cell to cell, very much like our desmosome. And very much like our desmosome, we would expect to find it in the upper mid-range of the lateral surface of our epithelial cell. If we had a transmembrane linking protein for our adherens junction that was an integrins protein, then we would expect that junction is linking the cell to extracellular matrix, such as a basement membrane, and that we would expect to find this particular junction on the basal surface of our epithelial cell, where it was making contact with that basement membrane. And that brings us to the end. I hope you found this tour of epithelial cell junctions beneficial, and I hope to see you next time. Thanks for watching.